tempted to totally rethink what I talk about today in light of some of the conversations I've had in the last few minutes and last night. And I won't do that entirely, but I will restructure how I begin a little bit. Which is to say that most of what I'm talking about today is about the idea that microbially, in the broadest sense of the word microbe, so to include things that are not visible, and even if they're macroscopic, that we tend not to see them, that microbially there's a kind of rift. And this is where did the stuff go? This is a sentiment that's not my own, it's a sentiment that comes from the work that's happening at the Rachel Carson Center, and I'll come back to this. That there's a rift between us and the life we don't see or pay attention to. And that rift has become more and more extreme, and as we've moved indoors, it's become almost permanently extreme. And one of the very few ways that we seem to know how to remedy this rift is by bringing some of those species back into our lives through baking, through fermenting, through incorporating these other lives into our daily stories. And so part of what I'll describe in that context is the scientific, but also what's always going on as I talk about the scientific is a human narrative of how we think about these other species and their stories and how they connect to our past. And so the microbial rift I'll talk about is biological. We're getting sick because we're separate from species we've long been with. But it's also metaphorical, it's human. There's a rift between us and the way we've long lived and the kinds of connections we've long had. So that's sort of the overview that I didn't have enough clarity on until yesterday and this morning. And now I'm going to get into some of the details, which will take us a long way away from sourdough for a little bit, and then we'll come back. And one of the directions it'll take us, this is in the context of this book that, let's see if this works, Claire. There we go. That came out last year about the life in houses, which is a natural history, but it's also this story of this change that's happening to us. And it's work that happens in the context of the reality that if you're a baby born in Europe or the US today, that you're likely to spend at least 23 and a half hours of the day indoors in some way or another. And so as we think about our future, we can change that. We can go outdoors more, but the general drift of our societies is toward more and more time with whatever species are indoors with us, whatever ecologies. And one part of the story that we figured out in the lab is that houses are actually much more full of life than we tend to think. And so if we think about a room like this one, if we think about a house like this one, and we go inside and we treat the individual parts of the home like little habitats, we can then sample those habitats, and then we can look at what's there. This is a bigger story, but the quick version would be that, for example, in the first 50 houses that we studied in Raleigh, North Carolina, we found more than 1,000 species of insects and their relatives in those 50 houses. When we talked to entomologists about what we might find, they thought we would find 20 or 30. And so there's a lot there. And those species are doing things. These are all species that are breaking down your house, breaking down your food. That's interesting. You never get to laugh among scientists, that. And you know, they can be quite beautiful. These are all the species that are eating those species. So if you didn't like the first ones, you should like these. This is a wasp that I love. Its only job in life is to lay its eggs inside the egg cases of cockroaches. They then eat the baby cockroaches alive and then fly away. People tend to like that one. We can also study smaller life in houses. So we can take a swab of dust, like we might right here. And when we do that, we can then decode the DNA in that swab of dust, or we can grow the species in the dust. And as we've swabbed houses, we've found many species of archaeans. This is a life, it's a kingdom of life or a division of life as different from bacteria as from anything else. This ancient division of life. When I was a graduate student, which is not so long ago, my daughter would say it was way back in the 1900s, which is not even true, but anyway. This is the number of species of archaeans known from Earth. This is the number of species of archaeans we found in houses. For fungi, this is the number of named fungus species in North America. That's the number of fungus species we found indoors in houses in North America, which also means many of these are not yet named. Bacteria, this is the number of mammal species on Earth, bird species on Earth. That's the number of bacteria species we found today indoors. And so it's a lot of life. Interestingly, fewer than 20 of those species are potentially dangerous. And in this room, there are probably 8,000 species of bacteria. Maybe 10 of those have been well studied. As somebody stands up in the back, the bacteria sort of waft from her toward the people nearby. And this is happening all the time. I didn't mean to make you sit back down, sorry. But when we think about the future, we often imagine this microbially rifted world. We imagine that what we want to create indoors is a world from which we remove all of these species. And what I would suggest is this is a terrible idea. It's this idea, right? It's the idea that in the future, nature is somewhere, but we've built these pods in which we live, and if we really need something to nature, we send somebody to go and get it, which includes our food, or everything else we need. But it's also fraught in the sense that this sort of teleological trajectory that we imagine from living in a chimpanzee nest to living in a city to living in this future world, it's not what it seems. I'll just do it the old-fashioned way. It's not what it seems in the sense that all of the worlds that we've created that are like this are not sterile. So all of the green buildings that we imagine we're sealing out are not sterile. And so the future in which we're totally separate from nature is not a good idea because we can't kill everything anyway. We can't achieve it. And this has become super clear in the context of studies that some of our collaborators did of the space station. So if you look at the International Space Station, it's the future home we've tended to the most with regard to trying to exclude other species. And our collaborators took the same swabs we used on Earth and they swabbed the space station, and what they found was that it was full of life that came up with the astronauts. It had dust mites, it had hundreds of species of fungi, thousands of species of bacteria, but there was something characteristic of those species. They were almost exclusively species associated with the falling apart of the bodies of the astronauts and with the deterioration of the actual materials of the space station. And so if we try to seal out everything, that's what we get, our own falling apart and the things that eat at what holds us in. And we can see this here. If we imagine a kind of map where points that are closer together are more similar in which species they have, this is what the space station looks like. It actually most closely resembles toilet seats on Earth. And so we can embark on that future, but this does not seem like where our hopes and dreams should go, right? But we're doing it all the time. This is a study by Deanna Beasley. I'll walk you through this figure. She studied daycares in North Carolina in the U.S. And what she found is that if you compared what was outside the window to what was inside the window, that outside the window, the vast majority of the bacteria were plant associated. So above 75%, look at that little bar in the middle, that's the mean. Going just a little bit inside the window, most of those plant-associated bacteria disappeared, and instead you had the falling apart of children and teachers. And this is what we do again and again. We can contrast this with chimpanzee nests. And I think what we think about chimpanzee nests, we think about them looking like what I just described at the space station, like the chimp version of a toilet. Well, except that Megan Temes, who's now at the University of California, San Diego, actually compared chimpanzee nests in western Tanzania to human homes, and this is what she found. In human homes, the proportion of microbes from bodies is around 35%-ish. In chimpanzee nests, it's barely noticeable. So the chimp nest is basically environmental associated microbial species. It's species from soil, from leaves, from the air. As we've moved indoors, we've excluded those species, and it's our falling apart. That's what's here with us right now. And then it's like those species that grow in the space station. These are all species cultured from a dishwasher. Many of these are the same species, most of these are fungi that are grown from the space station. They're able to withstand really harsh conditions. We make lots of these. We love to make lots of these. The Rachel Carson Center lunchroom has none of these. It's okay. Because I saw it and it doesn't look like anybody tried to clean that hard, so probably it's good. But one of the most conspicuous consequences that many, many studies are now revealing is that in sealing ourselves in, we favor body and extreme biodiversity and disfavor outdoor biodiversity, as I just mentioned, but also this makes us sick. This favors allergies, it favors asthma, it favors chronic inflammatory disease like inflammatory bowel, Crohn's disease. It's even associated with MS in ways that are quite complex. And so what we're really seeing is what people of my community call evolutionary mismatches, but one might also call microbial or ecological rift, which I actually think is a much better way to conceptualize this. And in essence, what's happening immunologically as all this goes on is that our immune systems, when we don't have normal exposures, get bored and they attack us. It's way more complex than that, but that's actually sufficient as a working model. The other thing to bear in mind as I talk about this is that we really can't kill all of this stuff. And so the space station is a model of that, but so too is what happens when resistant organisms evolve. And so when we really, really try to kill things, what we get are pesticide-resistant roaches, antibiotic-resistant bacteria, and this can happen very, very quickly. There's a beautiful recent experiment, which I really think is a humanist project more than it is a scientific experiment, by Michael Bain, by which I mean it's better than, in which Michael made this giant petri dish, and he put bacteria food down on it, and then at either end he introduced E. coli bacteria that were not resistant to any antibiotics. Just normal, safe E. coli. But then what he did was he played a trick on the bacteria where he put different concentrations of antibiotics in the petri dish. Now if the bacteria don't evolve resistance, what will happen is that as the bacteria get to the antibiotics, they'll all just die, and that's the end of the story. And so I'm actually going to show you what happens, and this is a video that takes place over 11 days. And so what you see are the bacteria growing. And then they hit a wall. That wall is the first level of antibiotics. And they bust through. When they bust through at that moment, it's because they've novelly evolved resistance. And the number of dots you see is how many different individual bacteria have independently evolved resistance. The other thing to keep in mind, two things. One is that this could happen in your house, right? Or in your body. The other is that this concentration of antibiotics in the middle should be the thing that destroys these in a way that the bacteria never get through. Somebody say, make them stop.
In now.
The way we make them stop is by not overusing antibiotics and having other bacteria present with which these bacteria must compete. If there were other bacteria on this Petri plate, the E. coli would not do well because E. coli are actually not very good competitors. But in the absence of the rest of life, they do this just fine. So in trying to kill everything, we not only disfavor what we need, we also favor the very worst. And we do this all the time. I've not checked in the Rachel Carson Center bathrooms yet, but check and see if this compound is in the soap. If it is, it's doing the exact same thing on your hand every time you use that soap. It might not be, and then it was too dramatic for no good reason. And so I've thought about this in the context of many different species we bring into homes, many different decisions we make about the future. But the thing I got to when I was writing the book was at the end thinking about how do we think about a more successful future? What's a better vision for how we do this? And I talked to many architects thinking there'd be some architectural solution, that somebody was thinking about how do you garden a home? How do you favor the species around us that we need? And there are architects who are interested, but there's not a solution yet that's being engineered at a scale that's going to affect people in the next 10 or 20 years. But where I started to see solutions was when I started to talk to bakers. We did a sourdough experiment that I can come back to later. We brought a bunch of bakers together from around the world to Belgium. And it was separate from this housework because the goal was really to figure out, when the bakers baked bread, how much of the flavor in the bread was from the microbes on the hands of the bakers. And so they each made the same sourdough starter, same ingredients. What varied were their hands and their bakeries. They then came together. We had to stop them from adding extra ingredients to the breads that they all wanted to add, like poppy seeds or garlic. The bakers kept pulling things out of their pocket. But in the end, it worked. So we got them all to make the same starter, all to make the same bread. And we had the breads tasted like you did here today. And then we also studied which microbes were in the sourdough starters. And what we found was each baker contributed some of the microbes in their starter from their own body. That was one piece of it. And so Matthew's family story is literally embedded in some way or another microbially in these starters. How much? Hard to say, but almost certainly there are individual genes that are present from the microbes of that story, that family. The other thing, though, which we didn't anticipate, was that when we studied the baker's hands, if we hold your hands up for a second. So right now you have two things going on. One is that there's really funky microbial things going on in armpits, and you're all sensing that now. That's a separate, very interesting story. The wiggling was, but the second is your hands on average have about 3% lactobacillus bacteria, maximum. The bakers had up to 70% lactobacillus bacteria, which meant that the way they had lived their lives ecologically had changed who they were microbially. And this, to me, is a kind of solution. That if we can imagine ways to live our daily lives in which we are gardening species that benefit us, it changes us. It changes us in a way that's persistent. And I think that the traditional knowledge embedded in home baking offers us many insights with regard to how we might do that. Now, the challenge is that, for the most part, scientists have neglected everything involved in the traditional knowledge associated with home baking and fermenting. Things have happened, there's science that's been done, but for example, there's not a single study that details which microbes are present in sourdough through time on multiple flour types, as an example. But we also don't know the history of fermentation. We have no idea when fermentation starts. It may be as long ago as 100,000 years ago. And so all of these things are neglected, but part of our potential future. And how am I on time now? What do I have before we get to questions?
Yeah, and we have some more minutes.
Some roominess?
Some more minutes, yeah.
Some more minutes and roominess. That's perfect. So what I'll quickly transition to then is how we've started to try to understand some of what's going on in that home baking context. And just for starters, what's happening with it, that was not meant to be a pun, but it was good. Starters are remarkably simple when you initiate them. With Matthew's starter, you worked with a starter that already exists. You just used it. But were you to make one newly, you would add flour and water, and from somewhere bacteria and yeast would come. The bacteria would produce acid, the yeast would produce carbon dioxide, and extra flavors, and so the bread would rise. The dough would rise. And then you have associated aromas, and you can do this around the world and it works. And so it's a pretty amazing thing. We can talk about how we might have discovered this in the first place thousands of years ago, and there's some interesting things. But what we've started to do in the lab, and this is work led by Aaron McKinney along with others, is to work with teachers and students and families around the world to start to study what's going on with starters. And so, for example, we have a project where we had people send in their sourdough starters, many of which have stories like Matthew's, stories of family and history, most of which are named. And then we began to study those starters to figure out what's going on with them biologically. And one of the things that we see is that most of the starters we see are a single kind of yeast. And so each line is an individual starter. But there are a lot of starters over here that have different yeasts. For the bread industry, these starters don't exist. These are different. Something different is going on. And you see the different colors mean there are many different kinds of yeasts. The same thing, and they're beautiful, is going on with bacteria, but there's even more variation. Most of the bacteria are supposed to be this red species, Lactobacillus brevis, or this pink one, Lactobacillus sanfrancensis, which people from San Francisco claim is their own, except that it's actually far more common in France, but we won't talk about that. But what you also see is that there are many other kinds of lactic acid bacteria. So there's a lot going on, and the variation we see from starter to starter falls into clusters. There are sort of these syndromes of microbes, different basic kind of types of microbes in starters. And so we can study these different types and see if they behave differently. And I'll just very quickly walk through this. Aaron and team have found that the different types of sourdough clusters, these different communities, they make the dough rise at different rates. And so once we hold constant the art of working with the starter and baking, which seems to vary here, there are different intrinsic rates of dough rise with different microbes. We can also do this fancy thing where we identify the aromas in the different starters, and do this amazing thing where an expert bread sniffer then simultaneously smells what the machine is smelling. And so do all these things to then... What a job, huh? But she sometimes has to smell things like, well, bad things. And so then we can identify for each starter which compounds are there, which aroma characteristics she identifies, and we can start to break those down. And we can talk more about them, but these are the kinds of things you might or might not have smelled in these breads. That some of the first one doesn't sound good, but I think probably actually is tasty. Cheesy, fecal, fatty acid sour, acetic acid sour, green apple, alcohol. But different people like the different breads. There's really complex biochemistry. But the different clusters consistently had different flavors. But here's the thing. We can only explain about 10% of why the clusters exist. Why Matthew's starter is different from another starter. Some of it has to do with temperature. If you keep your starter in warmer conditions, you favor body-associated lactobacillus because it's more like body temperature. If you keep your starter in colder conditions, you favor plant-associated lactobacillus. They have different metabolisms, they're different. Biogeographic region also matters. There's a yeast we only saw in starters from Australia. But the other part is clearly the art and the traditional knowledge of baking and making the sourdough, which I would estimate that we're at this point don't understand 90% of it. And many of the questions we need to think about to address that are history and humanities questions. And so these are just a few I was thinking about last night. Each of the clusters have histories. What are those histories? How do we think about them? Those are embedded in human cultures. They're also part of the broader, largely unresolved history of starters more generally, which is part of the even broader, even more unresolved history of fermented foods. And this history is embedded in traditional knowledge in daily life, but tends to be ignored by ecologists and evolutionary biologists, and also ethnographers, and I think most historians tend to ignore these kinds of histories in ways that relate to what we know about the science. Fermentation's a magic in our collective stories, a magic in origin stories, and even a magic that may have been key in human evolution, but it's a magic we haven't studied. We can talk about why it might have been unstudied, but that hasn't really been done that much either. And this is a garden of the past, but I also think a gardening we will need ever more in the future. And so I think also an amazing opportunity to bring together physics and chemistry and history and art and poetry to think about what the home should mean in light of all of these gardening things in the future. If there's time, there are some biological mysteries that are really central to what's going on with these starters that I think are very ancient. But I'm going to close there because I actually really want to have time for some good discussion. And so I'll stop there. I will say that we would love to do more of this kind of work in Germany. And so if there are ways to partner with any and all of you to think about interventions and experiments in Germany, that would be great. I think we have to do another thing next, and so I will leave this story of a starter that came to us. This is just an email that came with the starter up while we make the transition to the next thing. But thank you very much, and I look forward to the next part of this.